Laurence Zitvogel, professeur at Université Paris-Saclay, Gustave Roussy Cancer Center, and INSERM. Thanks to the organizers for giving me this opportunity to contribute to this educational session devoted to the concept basis of breast cancer immunosurveillance. We will successfully review through these following chapters. Let's start with a description of the conceptual basis of breast cancer immunosurveillance. In 2000, cancer was reviewed as a cell-autonomous disease resulting from perturbation in cell signaling, apoptosis, and angiogenesis. From a genetic and then epigenetic disease, cancer was progressively conceived as a chronic inflammatory process resulting in local and systemic immunosuppression. The cancer immunity cycle stems from the capacity of dendritic cells to sense and capture dying tumor cells and then migrate to tumor-draining lymph node to present antigens in MHC class 1 and class 2 molecules to educate T lymphocytes. T cells get ignited through the T cell receptor as well as the co-stimulatory signaling pathways, allowing trafficking to the tumor lesions. There, they operate in an MHC-dependent recognition pattern to eliminate cancer cells. However, mounting T cell responses is not always effective. The threshold, the duration, and the intensity of immune response will vary in individuals according to host and cancer genetics, comorbidities, co-medications, environmental factors, including the gut microbiota, that all impact on the proficiency of the cancer immunity cycle. All cancers are associated with somatic mutations. More than 5 million mutations were extracted from 7,000 cancers, leading to the description of 20 distinct mutational signatures. We learn that the T cell-based immune system frequently responds to the neoantigens that arise as a consequence of this DNA damage. Recognition of neoantigens appears an important driver of the clinical activity of cancer immunotherapies. However, breast cancer is not a prototypical histological type associated with a high tumor mutational burden. Besides their antigen specificity, T lymphocytes receive co-stimulatory signals from their tissue environment that greatly influence their differentiation patterns. Basically, Th1, Tc1 cells produce interferon gamma, IL-2, TNF, and are effector cells leading to cytotoxicity and allowing a protective anti-cancer immune response, while all the other cells, Th2, Th22, etc., are not. Specifically, regulatory T cells characterized by the FOXP3 are gatekeepers of tissue homeostasis, preventing the elicitation of effector Th1 cells. In particular, follicular T helper cells that orchestrate B cell responses and the formation of active germinal centers and tertiary lymphoid organs are key players in breast cancers. Pioneering work from Villar-Gallo and Perus group showed that the quality of follicular T helper cells and tertiary lymphoid organs dictate the prognosis of breast cancers. The observation has been extended to multiple tumors amenable to PD-1 blockade. Interestingly, a novel subset of tissue-resident memory T cells that do not emigrate outside the breast has been identified to convey favorable prognosis and predict response to PD-1 blockade in triple-negative breast cancers. Immunotherapy or immunogenic cytotoxicants should be able to restore a proficient microenvironment dominated by a crosstalk between activated dendritic cells and NK cells, leading to effector CD8 T cytotoxic lymphocytes helped by follicular T helper cells and Th1 cells, coordinating tertiary lymphoid structures and providing new T cell repertoires. This ideal tumor microenvironment is characterized by a dense T cell infiltrate with Th1 transcription profile, expected to pave the way to the efficacy of immune checkpoint blockers and adoptive T cell therapy. This is characterizing about 30% of TNBC and 25% of HER2-positive breast cancers. However, most tumor microenvironments are cold. They are not hot, meaning that they are devoid of T-cell infiltration because of T-cell exclusion, T-cell exhaustion, or no T-cell priming at the first place. These three circumstances predict resistance to direct immunostimulation. Hormone receptor breast cancers fall into this category. T-cell exhaustion is characterized by the overexpression of inhibitory receptors at the T-cell surface, such as CTLA4 and PD-1, that recognize for the latter the ligands PD-L1 and PD-L2 that are expressed at the tumor cell surface and compromise the effector phase of the immune response. More than 6 therapeutic antibodies targeting the immune checkpoints PD-1 and PD-L1 are currently EMA and the FDA approved to prevent T-cell exhaustion. Let's switch to breast cancer immune score to guide prognosis and therapy. The quantity and quality of T-cell density is assessed on hematoxylin eosin stained tumor tissue sections. They can be observed within the stroma or the tumor nest. A standardized method for TIL density evaluation has been provided by the International Immuno-Oncology Biomarker Working Group. It is recommended to assess the percentage of stromal TILs occupying intra-tumor stroma. Breast cancer TILs predict favorable prognosis in early breast cancers and responses to neoadjuvant chemotherapy and HER2-based therapies. The Keynote 119 shows that TIL superior to 5% predicted the response to pembrolizumab. Similar results were obtained in neoadjuvant trials with immune checkpoint inhibitors in triple-negative breast cancers, namely Keynote 173, NeoTRIP, and Guépard Nuevo. Of note, TIL assessment is not introduced yet into the routine clinical and pathological practice. It is being integrated into biomarker discovery studies associated with immunotherapy trials as a low-cost parameter, which can rapidly give an estimation of the tumor immune profile. In addition to TIL enumeration, TIL gene expression can help stratifying the subset of TNBC patients responding to carboplatin-based neoadjuvant regimen. Many factors influence the response of breast cancer to PD-1, PD-L1-based immunotherapies. PD-L1 expression and TIL densities are the main drivers and predictors of response to PD-1 blockade. PD-L1 expression is conditioned by TIL densities, interferon gamma release by those TILs, and the genetic makeup of cancers. For instance, tumors activating the beta-catenin Wnt pathway or presenting with PTEN loss will have increased PD-L1 expression. The take-home message is that the pan-tumor T-cell inflamed gene expression pattern has been validated across 9 different cancers treated with anti-PD-1 antibodies. Indeed, the type 1 interferon fingerprint associated with response to PD-1 blockade will encompass T-cell exhaustion, type 1 interferon, NK, and antigen presentation pathways-related genes. While 30% of TNBC respond to PD-1 blockade, hormone receptor breast cancer are left behind because they fail to express PD-L1 and to be invaded by tumor-infiltrated lymphocytes. Instead, preclinical studies using progesterone-induced luminal B breast cancers have shown that hormone receptor breast cancers may be readily immunoedited already by NK and CD8 cells during natural or tamoxifen-induced immunosurveillance. Indeed, such tumors contain high levels of regulatory T cells and are more controlled by NK cells than T lymphocytes. Breast cancer cells are amenable to immunogenic cell death when exposed to cytotoxic agents, paving the way to TIL infiltration and immunoediting. Successful over the past decade, our team showed that, followed by others, that successful chemo and radiotherapy should be considered de facto as immunotherapies because they induce a type of cell death and a type of cell stress before death that elicit long-term protective immunity. In this mouse tumor model, treated with anthracyclines, the efficacy of doxorubicin was abolished in the absence of T lymphocytes, suggesting that an intact immune system composed of a functional adaptive T-cell immune response is mandatory for the full-fledged efficacy of cytotoxic agents. Indeed, we showed across several transplantable and orthotopic tumor models that CD8 T cells, interferon gamma and its receptor, IL-1 receptor, toll-like receptor 4, and formyl peptide receptor 1 were all mandatory for the perfect efficacy of anthracyclines or radiotherapy. The immunogenicity of tumor cells relies on both antigenicity and adjuvanticity. While anthracycline did not modulate the surface immunopeptidome, we showed that they profoundly influence the adjuvanticity, meaning the expression or release of ligands for pattern recognition receptors common to human and non-mammalian model organisms. Where are such adjuvants coming from? Cell stress preceding cell death is critical for the immunological outcome during chemotherapy. Each cellular stress can generate the expression of the release of toll-like receptor agonist. Indeed, ER stress response triggers calreticulin exposure, which is mandatory for phagocytosis of dead bodies, for instance. Activation of the autophagic machinery will lead to ATP release that causes dendritic cell recruitment and IL-1 beta secretion for innate immune triggering. Cell death per se generates the extracellular secretion of the nuclear IMGB1 and annexin A1, engaging toll-like receptor 4 and formyl peptide receptor 1 respectively, which mediate dendritic cell dead corpse close interaction and activation for antigen processing and presentation. Viral mimicry is occurring through ignition of the type 1 interferon signaling pathway in tumor cells and culminates in CXCL10 release to cause T-cell chemoattraction. Lastly, only distinct therapy can trigger these factors, namely anthracycline, actinomycins, D, taxanes, X-rays, ALK inhibitors, cyclophosphamide, and oxaliplatinum. Therefore, any breast cancer that is defective in the expression of any of these ligands will not be able to die in an immunogenic manner, precluding long-term benefit to cytotoxicants. Hence, breast cancer defective in nuclear HMGB1 expression, as you can see here in immunohistochemistry, or disabled for autophagy induction, as shown by the LC3 staining, will relapse earlier than breast cancer proficient in these immunogenic cell death markers. We also showed that breast cancer deficient in immunogenic cell death hallmarks have also low T-cell infiltrates and higher invasion with immunosuppressive M2-like macrophages. As exemplified by many teams, type 1 interferon signature in tumor beds centered around CXCL10 metagene predicts response to neoadjuvant anthracycline-based chemotherapy and can be diagnosed with anti-MX1 antibodies in immunohistochemistry. Consequently, as observed for pathogen infection, patients harboring single nucleotide polymorphism residing in pattern recognition receptors sensing immunogenic cell death hallmarks, such as toll-like receptor 4 or formyl peptide receptor 1, will have reduced clinical benefit to cytotoxic therapies. ICD is an important biological step prior to the initiation of immune checkpoint inhibition, as shown in this immunity paper. In a cold pulmonary tumor induced by oncogenes and devoid of TILs, the combination of oxaliplatinum plus cyclophosphamide could elicit the ICD hallmarks, and this is a prerequisite for the chemoattraction of TILs and the success of immune checkpoint inhibition after oxaliplatinum and cyclophosphamide. This paper was followed by the in vivo proof of concept in the tonic trial in TNBC treated with anthracycline and boosted with PD-1 blockade. Similar exemplification were brought up in metastatic breast cancer treated with immunogenic paclitaxel and anti-PD-L1 antibodies, and in lung cancer treated with radiotherapy and CTLA-4 blockade. The GIADA trial illustrated the implementation of this concept of combining immunogenic cell death inducers with immunotherapy in neoadjuvant settings of luminal B breast cancers. In fact, most of the drugs recently approved in breast cancers, such as HER2, PARP, or CDK4/6 inhibitors or hormonal therapies, can promote ICD hallmarks and synergize with immune checkpoint inhibition in preclinical tumor models. The concept of immunogenic cell death can be extended to antibody-drug conjugate, delivering the ICD-mediating agent in the lysosomal compartments of tumor cells. HER2 and TROP2 redirected deruxtecan is an excellent example illustrating this concept, paving the way to combinatorial regimen with PD-1 blockade. What is the impact of gut microbiota on breast cancer management? Surprisingly, tumors are not sterile, and intratumoral bacteria can deliver toxins or DNA damage inducers or immunosuppressive molecules maintaining inflammation and DNA repair deficiencies. In particular, breast cancer represents the most bacteria-invaded cancer type. In contrast, we showed that gut microbes can participate in the peripheral and tumor immune tonus with immunogenic commensals mediating antigenic and adjuvant function in play during PD-1 blockade. Gut microbiota taxonomic composition at diagnosis of early breast cancer is surprisingly associated with TNM staging pre and post chemotherapy. Moreover, chemotherapy has a power to change the gut microbiota taxonomic composition towards a beneficial composition for protective effects of the cytotoxic compound. For instance, Egertella lanta is associated with higher staging at diagnosis, but is underrepresented post-chemotherapy. Thanks to the avatar gut humanized mouse models, we can demonstrate that the human microbiota of breast cancer patients can colonize mice in a different way than stools from healthy volunteers and influence their response to cyclophosphamide in a breast cancer tumor model. What are the consequences of all these concepts in breast cancer prevention? For instance, the formal peptide receptor 1 receptor for annexin A1, which is involved in the dendritic cell dead corpse interaction, harbors a genetic polymorphism present in 4% of healthy volunteers in a homozygous status, but 25% in an heterozygous form. This polymorphism accelerates breast cancer incidence by 5 years, not only in breast cancer, but also in colon cancers, in several independent cohorts in multivariate analysis. Hence, prospective validation is warranted to document its relevance in breast cancer prophylaxis. Likewise, mice harboring a defective formal peptide receptor 1 annexin A1 pathway can be compensated by toll-like receptor ligands, such as the poly-IC LC, that allows restoration of dendritic cell corpse interaction in a T-cell dependent manner, circumventing primary resistance to chemotherapy. Lifestyle and nutritional interventions may play a role in dictating the peripheral immune tonus. Vitamin B3 or nicotinamide, which are active in preventing virus-induced skin carcinoma, prove very useful to boost T and NK cells during breast cancer hormonal receptor positive. Nicotinamide resets a type 1 interferon fingerprint, leading to T-cell infiltrates in cold hormone receptor positive luminal B cancers induced by medroxyprogesterone in mice. In conclusion, luminal breast cancers may benefit from new immunological armamentarium based on NK cells and or the Treg elimination, as well as restoring gut microbiota abiosis. HER2 breast cancer may benefit from novel immunotherapeutic approach or bispecific antibodies or ADC version of immunogenic cell death inducers. Triple-negative breast cancer may benefit from better stratification and novel immune checkpoint inhibitors. The landscape of biomarkers will flare up in breast cancer in the near future to better stratify treatments. Thank you.